Hello, everybody. Good morning, good afternoon, or good evening, depending on where you are. It's a tremendous pleasure for me to open the town hall today, where Aviv and I will be updating you on recent progress and new developments, and then taking questions at the end. So I hope everybody can see my screen and my pointer. Any issues, please put it in the chat. So the agenda for today is to summarize progress and future goals, then to talk about Atlas integration and cell annotation. And we have with us here today as panelists, Chloe Villani and Fabian Theis, specifically for the second point. Then we'll update on the white paper version 2.0 new release, and also a publication bundle that we're aiming towards for next year for the HCA community. Heads up on events for the coming year, and then at the end, the question and answer, ask me anything. So the 2022 has been a really amazing year for the Human Cell Atlas, kind of rebounding from the pandemic. We're now at a point where cumulatively over the years and across the entire community, we've got more than 1,000 million single cells or nuclei covering many, many different tissues across the entire body. So this is really an exciting time for the Human Cell Atlas. We had our first cross-tissue publication bundle, kind of a mini bundle of 3 papers in an issue of Science, with a 4th paper published at the same time online. And all of those 4 papers were cross-tissue papers. So they were addressing the similarities and differences of cells across tissues. One of the main focuses was the immune system and how do cells compare from the same compartment across different tissues. So that was incredibly exciting. And that little mini bundle, which evolved sort of spontaneously, got a lot of attention and coverage, which was nice for the project. We also went back to hybrid meetings with in-person conferences and coupled with online in Vienna, Austria, which was hosted by Christoph Bock and colleagues in Austria, which was lovely. We had a HCA Asia meeting very recently in Bangkok, Thailand, hosted by colleagues at Mahidol University, Varan Charansion and Ponpan and others, which was really, really fantastic for the HCA Asia, and also had the online component. And then we had a first in-person pediatric and development meeting in Paris, which was fantastic for the HCA development community and the nascent kind of growing pediatric community. And so the entire membership now encompasses over 2,500 members from over 80 countries worldwide. So that's pretty amazing, and very, very exciting to have that global membership, and that's continuing to grow, in particular with the geographic communities in Asia, Latin America, and Africa, and the regional networks in those parts of the world. Another exciting development is that the biological networks are now sort of crystallizing their roadmaps for the next phase of the Human Cell Atlas. And there have been publications from many of those networks, including the lung biological network, the development network that had a nice roadmap paper in Nature. The gut biological network is in the final revision, in the revision phase for one in a Nature Reviews journal. And so these are papers that sort of lay out the plan for the next phase of the Human Cell Atlas for each biological network. So how do we want to complete the gut cell atlas, the lung, the airways, the development cell atlas, kidney, et cetera, et cetera. And so that's a kind of impetus for those networks to grow into the next phase. We've also had the launch of CZI awards on ancestry, and also funding for pediatric work from requests for applications from the Chan Zuckerberg Initiative. And those grants will kind of be really, really fantastic in boosting the pediatric cell atlas, and also in boosting the an increase in sampling across diverse samples for the Human Cell Atlas. So we're incredibly pleased and grateful for the partnership with the Chan Zuckerberg Initiative on those fronts. And we've also had multi-year support for the HCA Executive Office. And that's just important because the staff, and some of them are here, who support us, 2,500 scientists across the world, provide that backbone and that support for meetings, for these kinds of events and so on. So we have here with us today, Christine Schwenk, Jenny Wu, John Randall, and others from the Executive Office, and they're instrumental in making all of these events happen and the project move forwards. So as I mentioned, we're now at the sort of 100 million suspension cell nuclear mark, of course, and that's getting us towards a really respectable first draft of the cells in dozens of tissues in our body. And looking forward to the roadmaps for the next phase, we'll be looking at a more comprehensive atlas with a larger number of suspension cell nuclear data, including multi-omic data, and also much more spatial data, of course. So to kind of visualize this in a graphical sense, and these are, log scale kind of axis, we've got on the order of tens of thousands of individual samples, tens of individual donors, similar sort of order of magnitude samples, and then hundreds of millions of cells. So this is a really rapid growth in data. And in sort of summarizing the views from the atlases, you can see there are many different tissues and networks that have kind of contributed these hundreds of millions of cells, and they include also COVID, 12 million cells from COVID infected donors. So looking forward to the next year or two, what we hope is that we'll move forward on integration of an initial set of tissue atlases. So that's focusing on not just the integration of the lung, which is on bioRxiv and under revision, manuscript under revision, where almost 450 samples from the airways have been integrated, as a collaborative effort. Fabian, who's here, is involved in this. Really, the analysis working group with the lung bionetwork have kind of come together to integrate all of this data and provide a gold standard reference of suspension cell data for the airways. And what we're hoping to do is to push forward integrations of other tissues in a similar manner, so that there are integrated gold standards. And this is really, I want to emphasize both a computational challenge as well as a biomedical kind of scholarship challenge in integrating the data, annotating it and interpreting it, and providing it ultimately through databases and data portals for use of the community, because the Human Cell Atlas is now in that phase where there's so much data that we need to start assembling it and serving it for the community in a way that's user friendly. And one element of that is of course cell annotation. We've got Chloe here for that, for discussion of that later. On the data coordination platform side, implementation of managed access and data access committees for ease of combination of fully open access public data with controlled access, the managed access data from living donors is a significant advance for the data coordination platform. And then in terms of, again, having sort of democratized user-friendly access to Human Cell Atlas data, we need to work on integration of the DCP with data portals that simplify querying, use, and interaction with the data. Another goal is expansion of training programs and courses across the world, basically, especially underrepresented regions. So looking forward to the future, we want to integrate remaining tissue atlases. And so I mentioned that with Fabian and the Lung Bionetwork and Malte Lücken and many others, there's been initial integration of the airways. there's a team of 12 postdocs spread across multiple groups working together on integration of other tissues where there's a wealth of data, and that includes gastrointestinal tract, kidney, brain, et cetera, and so pushing forwards on that integration and providing those gold standard references is an important aim for the next 18 months roughly. Evaluation of potential use scenarios and strategies for querying is again related to providing access to the data in a user-friendly way, which will ultimately represent the release of the first phase of the Human Cell Atlas in an online user-friendly kind of portal form. So just to summarize, what we see sort of in the longer term, so is this, the sort of fuller version of the Human Cell Atlas with extensive cross-tissue integration. Obviously, lots and lots of spatial data is going to kind of be emerging, and we'll have that spatial phase basically for all of the tissues for the Human Cell Atlas within the coming years, and we kind of expect that wave to sort of increase exponentially. We'll have an increasing scope in terms of genetic diversity, ancestry diversity, and so on, and that's really fantastic and exciting. Which and all of those data sets will of course impact the medical relevance and the clinical utility of the Human Cell Atlas. And in terms of long-term maintenance for the data access, data portals, and so on, we need a long-term strategy so that the Human Cell Atlas can be of use to humanity as a molecular map of the human body for the longer term, the next decade at the very least. And this really brings me to the next section, which will be dealing with Atlas integration, cell annotation, and then looking forward to the white paper and the publication bundles. And for that, I will hand over to Aviv, who is here, and I'll advance the slides if you tell me to, Aviv.
Great. Thanks, Sarah. So Sarah actually started touching on, I think, what is one of our greatest opportunities and challenges. There's 100 million cells or so. They come from almost 10,000 individuals. This is a massive integration effort, and that's actually just the first draft. So in the last year or so, we have really turned a lot of attention in this direction, and it's now in a full-fledged mode. So if we go to the next slide. Great. There's multiple different data platforms and projects, and I think sometimes people get a little bit confused between them. So I'll take a moment to try and give a sense of where they are. So first, I saw even a question in the Q&A, where's the data? So not all 100 million cells are already public data, because that is actually based on a census of all the data that people in HCA member labs have been collecting. And some of this data is, from yesterday and has not yet been QC'd, and some of this data has already been published 3 years ago. So there's quite a range in that respect. But data that is ready to be out there is released in the Data Coordination Platform, or the DCP, with the important caveat that today we still cannot release raw controlled access data through the DCP. It's often available through other resources, and sometimes you have to approach the author directly, and that is one of the goals for the coming year that Sarah already highlighted for you. Next, there are portals, still previous slide. There are the portals, which are places where you can come and really engage with data in different ways. For example, you can visualize it, you can slice and dice it in different ways. And there's actually a whole universe of these kinds of portals of many different kinds. There's a cell by gene portal, there's a portal at the BEBI, there is the single cell portal, and each of them has different types of tools and capabilities. Their multiplicity is a big benefit. Third, I want to highlight the cell annotation platform. We're going to have a few dedicated slides on that, and Chloe will be on the panel later. This is a tool that really is made in order to allow the community to collectively annotate cells, because if cells are not annotated, it's very hard for us to really manage them. And then finally, there are computational processes, platforms, and approaches that allow us to integrate data together across different kinds of studies. So if we go to the next slide. Go to the next slide. This really describes how pieces get put together in the process of Atlas integration. This is an effort that is now being kicked off, and we're very grateful to generous support from the Chan Zuckerberg Initiative that is helping, has helped nucleate this effort. This effort is led by several groups working together, including Fabian Theis's, Sarah's, John Marioni, and myself as well, and specifically, it is led as a project lead by Malte Luken at the Helmholtz. And these make individual teams, each of which takes on a particular atlas in a particular tissue and works to bring the data that is available and relevant right now for that tissue or system and integrate this. So the first step of that is actually to define the scope and detect the data sets, identify the relevant metadata and process it, then perform in the phase one, really, the basic QC and analysis of the different batch factors that could be impacting the data, harmonizing the cell labels, annotating kind of the seed data set, benchmarking the integration that we see, integrating the data, and again, repeating processes like clustering, selecting markers, and based on that, re-annotating and performing downstream analysis. What's important to understand about this process is that it is actually highly iterative. Some of the steps, there may be initial steps like dataset selection, you select your datasets, then you move on to integrate, but in reality, new datasets arise, so even that piece gets iterated. But other steps are inherently iterative, for example, the annotation process. And they are not done in isolation or in a vacuum. They are done in tight collaboration, coordination, and iteration together with the biological network, both on an ongoing basis and through jamborees. And if we go to the next slide. Oh, that was a click, actually. You can see the selected, the previous slide. You can see the selected systems and tissues that the first effort is focused on. There's the brain, the kidney, the gut, the lungs, and actually the immune system as well, specifically PBMCs. You can see them. I think that's that little droplet of blood. And you can see the different process, and the eye. And you can see the different atlases are in different phases of this process. So for example, for the kidney and the brain, the gut, and the second version of the lung atlas, we're in dataset selection. For the 15 dataset, spanning about 10 million cells from 3,000 donors that were selected for data QC, that were selected for from PBMCs, they're already in data QCs and batch analyses. For the eye, there are 9 datasets and we're in data integration. And for the first version of the lung, that has already been released. So this kind of process will iterate itself and things will find themselves not just at different points along this path, but actually at multiple points simultaneously, as you can see for the lung. There could be one version that's advanced and a second version, a more elaborate version, coming behind it. Next slide, please. Doing this requires certain types of infrastructure, both code infrastructure and also tools that the community can use. So for example, tools for metadata harmonization are not just about computational tools, but they're also about having an actual metadata file that is harmonizable. And the same is true for cell annotations and labels. And in addition, you need the right kinds of tools to serve data to serve integrated atlases for users. And I already mentioned one of the portals, the Cell by Gene portal, which is, and that effort is in partnership with the CZI that actually develops this particular portal. If we go back to the next slide, I emphasized several times already the critical importance of annotations. I think this is something that we all really appreciate. If things don't have names, they're very difficult to work on. And if things have multiple names and we don't realize that we actually refer to the same entity, they can sow enormous confusion in the scientific community. And so actually 3 years ago, HCA recognized that cell annotation is going to be a really important challenge. In a partnership with Schmidt Futures, we obtained funding to actually allow us to develop an annotation platform, which would be a software tool that allows users to come together, annotate things, and also gradually connect and harmonize those annotations. And this is led currently by Chloe Villani, and again, with participation from multiple groups, including from Fabian Theis and from John Marioni. And so the first, and from Niels Golgenborg, my apologies, I almost forgot. And so one big first component of the cell annotation platform is to create, contribute, and view cell annotations. There's an interactive user interface that allows you both to look at annotations and explore them. So you can see the molecular signatures that are available for different public data sets. You can browse the existing annotations, and then you can add your own annotations you're using the browser and publish. For example, you can look at a label and say, I would call it by a different, maybe more elaborate name, or you can identify a new group of cells and give them a label that wasn't there before at all. And it allows the users to provide also the explanation and evidence that underlies their annotation. And then the annotation component has all the synonyms and categories and different lines of evidence. The second major component is that you have a collaborative space by which to annotate the cells that the biological networks can use. And so a user can be a viewer, they just look at an annotation, they can be an editor, and they can own the entire annotation setting, say, for a biological network. If you go to the next slide. Oh, and I should say the most important thing is then it took a while to develop this annotation platform, but it is now actually ready to use. Next slide, please. Now, some of the upcoming features, there will be a transfer UI. So you can take any data set in CAP in the cell annotation platform, and you can use it as a reference. And then the user would select the reference and a model that they're interested in, and map the annotation. And then the user can view the annotations once they've been mapped and accept, reject, or refine them. In the spring, we're going to have a real-time suggestions tool that would be able to do it based on text-based evidence and molecular evidence together. And in the fall, we should already have cell cards, which will be reference summaries. This is a mock-up from a couple of years ago that would really show you, here is where your annotated cell type is, here is where it is within a cell ontology, and here are all the features that go along with it. And this is a little similar to, the gene cards concept that has existed in genomics for several decades now. If you go to the next slide. And so this gives you a little bit of an overview of both where Atlas integration and annotation is in. These are things that would require engagement from many people in the community. We're both really grateful for the leadership of the teams that are leading it, but also for the community contributing to all of these efforts, and especially for cell annotations. Let me go very quickly also through some of the ways in which we see publications happening in 2023. The first component in the next slide is the publication of a revised or an updated version of our white paper. As you know, we've never published our white paper in a journal because we consider it a living, breathing document that reflects on an initiative that continues to change. And a lot has happened since 2017 when we last updated the white paper. And Sarah just gave the highlights of 2022, but there were many of the years before as well. And so we will have both new sections and expanded sections, including all the bionetwork roadmaps that Sarah already referred to, a much expanded section on the computational challenges and our data ecosystem, the different regional networks of HCA, something we're very proud of and didn't exist in 2017, and updates on our many achievements, as well as the plans and the governance that we have today, which is a little more elaborate than we had in 2017. We're in the process of still collecting and integrating the sections. Some of you in the audience are probably working on writing them, so please continue to coordinate with your co-authors, and we expect drafts to come in this month, and the integration just of those drafts is going to be a major lift. And if you have any questions or there's something you want to contribute, please email Jenny Rood, who's also on this call, at roodj@gene.com. Next slide, please. The second, and we think very important thing that is coming in 2023 is publication bundles for HCA. As you know, we ran a survey through our community on whether people would want to coordinate publications in more of a package format rather than as individual papers. There's hundreds of papers now that are HCA papers. You can find them on the website. We all read them and benefit from them. And we've always had this streaming mode where people in the community simply submit their papers, badge them as HCA. We have a publication committee that reviews them, and they move on. But we feel that we turned the corner when we had our cross-tissue atlases and published 3 of them in a coordinated way together. This was received really well by the community, and I think increased the impact of the papers, not just in visibility, but also really in their scientific impact, because people could compare and contrast approaches, where the insights are at, and what we have in this area. And so after surveying the community, we identified 3 major areas that we had real energy in terms of upcoming papers. One is papers around especially analytical methods. The second were papers around organ or system-specific atlas integration. There's many works like that and the data that goes along with them sometimes. And the third area of developmental biology and the developmental cell atlas, as well as other papers that are hard to just put into a bucket. And so we will be calling for preliminary abstracts by the end of this month, with the idea that within 3 to 6 months thereafter, or 4 to 6 months thereafter, depending on the topic, there will be already paper submissions. And we're going to follow a process that has been very successful for other initiatives. We're still in negotiation with journals, but we hope to follow a process that was successful for other initiatives, where people can submit in staggered ways, not necessarily all on the same deadline, and their papers get accepted and even published often in electronic format, but the print version comes out only after all the papers are together, and when there's kind of front matter like reviews or integrative pieces that really pull the entire package together. Next slide, please. The last topic to highlight is our activities and events anticipated in 2023. Some of these already have assigned dates, and some of them are coming up. We will have both virtual and in-person events. The virtual events give us the biggest scope, but there's something to in-person interactions that you just can't replace, as we've seen in Vienna and more recently in Paris for the developmental meeting. So Atlas Integration and CAP will have a virtual seminar in the context of our BioNetwork seminar series coming up in February. We will have several regional meetings, a meeting in Accra in Ghana, for a workshop on computational and experimental design in March. We will have a meeting, another workshop in Africa, non-virtual, again in Lagos in Nigeria in June, again on computational and experimental design. That's also part of our efforts from the Equity Working Group. And we will have a computational workshop in Latin America, in Chile in September, and the Asia annual meeting, which will be in December, and the date and location are still precisely TBD, as well as the Middle Eastern workshop, which I think is our first, again with the location TBD in December. In addition, from the non-regional, but the overall HCA, the BioNetwork seminar will include one on Atlas integration in February, a focus on technology in March, and additional programming will be forthcoming. We plan to continue these town halls virtually every 6 months. So the next one roughly will be in April. And I think I covered all of the ones that are planned, and of course, a general meeting next summer. And you can always track us at humancellatlas.org/events. Next slide, please. And that will be it. And so thanks for joining this part. And I think the rest is for our panel to take Q&As, and I'll turn it back to John and Ellen.
Well, thank you very much, Sarah and Aviv. That was really tremendous, really exciting updates throughout, and really looking forward to the rest of this webinar, which is really devoted to a Q&A with our wonderful panelists. To introduce myself, I'm John Randall. I work in the HCA Executive Office as the Chief Alliance Officer. The Executive Office for HCA, which has already been referred to by Sarah and Aviv, consists of about 10 professional staff, primarily based at the Sanger Institute in the UK, the Broad Institute in the US, and the Riken Institute in Japan. We are also excited to be expanding the Executive Office to Singapore, and we do have currently an open position available there. So if you know of any suitable candidates, please get in touch with anybody in the Executive Office, and we'd be happy to provide additional information. Today, we're very fortunate to have 3 additional members of the HCA community here to join Sarah and Aviv for the Q&A. They are Fabian Theis, who is a member of the HCA Analysis Working Group. He is a member of the board of directors of the US-based HCA nonprofit, HCA Incorporated, which really provides both financial support for the executive office, but also supports data compliance related to the handling of personal data as well. And then also he's a PI on the integration project, as has already been referred to by Aviv. Next, we have Chloe Villani from the Broad Institute, MGH, and Harvard Medical School. Like Fabian, she wears many HCA hats. She is a member of the HCA organizing committee. She is a co-coordinator of the Immune Biological Network, and she is the lead PI on the cell annotation program. And so we're pleased to have her with her. And then we have my colleague, Jenny Rood from the HCA Executive Office. Jenny is HCA's science writer and is leading the drafting process for the updated white paper that you have just heard about as well. I encourage people to enter questions in the Q&A. I see that we already have quite a number. I do want to ask you to use the Q&A field rather than the chat whenever possible, and then I will select questions to pose to the panelists. I want to start actually with the general question for any of you, actually of my own, and that is, what are some of the early lessons that you've seen from the integration and annotation efforts, either in the context of the formal programs or just in the context of the research networks that you're a part of? And I would just put this to anybody who would like to answer that question.
Maybe I'll start with two. I think from actually the effort of all the groups, I think one of the important lessons is how important it is not to just do it based on what works for one organ or system. So we have, Fabian has led a great effort in the lung, and it's been hugely successful, but the lung is not the brain. The central nervous system, I mean, the eye is very different than the rest of the brain. The kidney is not exactly the lung. So that's one part, that there is always this specialization, and that this specialization actually requires the expertise of the people who know these organs and tissues to do the right computational work. It's kind of a lesson we repeatedly learn. It's not that we don't know it, but it's always impressive when you see it. And at the same time, I would say, it's also inspiring to see how much can be learned across. That you do take things that you've done in one setting, and with the right modifications, they become very empowering for another setting, so you don't reinvent the wheel. That balancing act, I think, has always been substantial throughout HCA, and I think that's going to be very, very impactful for us going forward. The second and little thing, and I might hand that to Chloe, is I mean, even after many years of working with the immune system, it never ceases to amaze you how much of it there is. And the reason I say that is that one of the things that we really appreciate is how difficult it is sometimes to really do justice by the immune cells when you look at them through the lens of only the organ in which they are. And yet how huge it is when you try to think of them across everything. So again, one of these balancing acts, but it's something that really came to the fore when the team had to choose what to do exactly for the immunology component at first.
Sorry,
Yeah.
Chloe, go next. John, are you? Yeah.
Sure. Yeah, Chloe, do you want to add to that, please?
Yes, actually, it was a perfect introduction. I was going to say two lessons as well. Building on what Aviv just described, I think annotation strategies, they do need to be tailored to a different organ system. And there, aside from my favorite system, the immune system, there are a lot of organ systems to annotate. And there's some organ systems where we know very little about these cells, and some where we know a lot. And for some of these organ systems where we don't know as much, the only information that's out there is a cell ontology. And sometimes these terms are not even correct just yet, or we don't understand the relationship between the cells. And so that's why we brought together a large team to develop the cell annotation platform to empower all of you to annotate your cells. So with that, I want to make a little plug for the cell annotation platform. Aviv did mention during a presentation that it took a while to develop, and many of you may not have heard about it because initially the team had to develop the product, the software. And when we say it's currently ready and live, it's important for all of you to know this is not the final product. We're at a stage now where we really need your feedback. This is meant to be for all of you, right? It's meant to serve the HCA community. So as you upload your data, you try to annotate yourself. We want to hear from you. We want the feedback. Let us help you. And so as you try to figure out, the features, and you may realize you need some tailored features to help annotate your cells, do reach out to the team. When you get to the portal, there's a question mark that you can click on and send your request with questions, and we're going to circle back and meet with you. We're also going to be doing some different reaching out activities and Jamboree in early 2023, so we can help all the different bio-network tailor our tools. Thank you. Fabian.
Thank you. Chloe, do you want to add to that, please?
Yes, actually, it was a perfect introduction. I was going to say two lessons as well. Building on what Aviv just described, I think annotation strategies, they do need to be tailored to a different organ system. And there, aside from my favorite system, the immune system, there are a lot of organ systems to annotate. And some where we know very little about these cells. And so as you try to figure out, the features, and you may realize you need some tailored features to help annotate your cells, do reach out to the team. When you get to the portal, there's a question mark that you can click on and send your request with questions, and we're going to circle back and meet with you. We're also going to be doing some different reaching out activities and Jamboree in early 2023, so we can help all the different bio-network tailor our tools.
Yeah, thanks for nice introduction. And of course, I think this discussion of how we can now use atlases, that's sort of where we're getting, right? So we're building these bigger ones and now we're mapping, I think has been something that not only general HCA, but also in particular the computational community has been getting quite a bit of focus on. I think, Aviv, you kind of hinted it already. There's no one size fits all. In machine learning, actually, there's this no free lunch theorem that tells you there's no one universal model to conquer it all. It's been usually written up in the supervised case, but for us, from an unsupervised setting, it's quite similar. So I think what I learned a little bit from all of these sort of efforts across maybe our usual comfort zone of the people sort of the top we've been mostly talking is that one, the idea of mapping on top, which is sort of a bit related, maybe even quite similar to using, is becoming more and more exciting and hot, and it's not as trivial as we originally thought. So you just look up yourself, but sort of adapting and seeing what happens. And as outcome of this is that quite a lot of exciting biology just by sort of increasing scale and cool atlases will be delivered. But also, and this is a perspective I want to take here on from the computational side, there's a whole fun of new opportunities and potentially also challenges that we find, wonderful method adaptations and maybe also developments that we need. How can we do better integration mapping? Could we include time points in a more consistent fashion? How do we deal with the different modalities? That's also always things that, the community has been hinting at those past 3 and 4 years, but with this scale going up, with potential now going cross organs and so on, I think this is exciting. So my last point would be that I think these atlases could turn into quite exciting challenges on the computational side as well, and benchmarks. And so I think the input here of the community is really needed, and we would really like to get your feedback, contributions to that. You saw a whole bunch of, for example, this largest machine learning conference, NeurIPS, 2 challenges coming up, perturbation mapping. I think exciting ideas there and a lot of opportunities from the HCA community.
Thank you very much. I want to pick up a little bit on that with one of the questions in the chat about what you feel is sort of the biggest technical bottleneck for HCA right now. And also, do you feel like we have enough data right now to give us a complete view of cell type characteristics, or do we really need more data? Aviv, would you want to start with that?
So I'll start with the second part. I think the answer to the second question, do we have enough data, is it depends. And it really depends on the particular organ or system. I think in some of them, we're probably sampled quite well, and in others, we're not sampled as well, even across the board. And so the human brain being a great example of something where we know upfront we're undersampled. There's a beautiful data set of 3 million cells that's just been released from the Brain Initiative. But BCAN, the real initiative, the brain cell atlas effort, he's really just launching in the human. Until now, most of his focus was the mouse. So there we know we're under, but that's totally fine. We also know how to get there to get to the right place. In other tissues, I think we're in a much better shape. But even in those, as others actually commented in the question and answer, there is a lot of heterogeneity in organs, in large organs. And some of it is also very tied later on to the heterogeneity of disease in the organ, like the right side and the left side of the organ will behave differently. This part and that part of the lung will behave differently, and so on. And we sometimes need to go to pretty precise spatial sampling in order to get the particular organization of cells in those tissues. Whereas in many other large organs, there's a lot of repeatability, and just doing it again and again and again in order to cover the full volume is pretty much wasteful. And part of what is useful about the Atlas integration with the rich metadata is that it can allow us in a statistically reasonable way to actually identify where are we probably done in terms of the rarity of cells that we covered and so on, and where are we not. And done is never done in biology. It's only done up to a certain proportion. So by analogy to say human genetics, a new person is born every second, and that person has unique genetics, and we don't have it yet. And they have some private variants that no other human on the planet has. The genome is big enough and the options are big enough, so that just happens. But that's okay. What we actually say is, we know the human genetic variation of this ancestral group up to that level of rarity. And that's really what we're looking for, similarly by analogy in cells. And in that, we still have a long way to go for diverse populations, for some organs, for particular parts of organs. And the Atlas integration is going to help us understand what's the gap between where we are today and where we want to be. But as we've said since the beginning of this journey, a partial map is actually very useful. So that's one of it. The question was also on technical challenges. I think there's several. Fabian already touched on some of them, so I don't want to repeat those. I think experimentally, one of the questions asked whether the sparsity of sampling in the cells is a big concern. I think our community has by now concluded that it's probably not the biggest concern. It's always nice to have more, but we're actually sampling a lot of cells, and in each cell we sample randomly. So as a distribution, we actually estimate them quite well. It's a more statistical view rather than an individual cell view, actually, of cells. But I think one of our biggest challenges is going to be both deploying experimentally spatial techniques that are sufficiently successful for us at the scale of the atlas, rather than the scale of labs doing a little bit, doing a much bigger effort, and combining those data with the cellular level data, not just as a technical exercise, but also to yield the right biological insights. And I believe that these are still early days. Surprisingly so, we actually thought, I think that branch will be maybe a little more expensive than it was, but now it's really picking up steam, as Sarah already referred.
Great, thank you. There are a couple of questions, a couple of related questions about the interface between annotation and integration. Sarah, did you have something you wanted to add? Sorry. That last question.
No, I'm happy to move on to that, to the next. I'm happy to, yeah, I mean, I think that the interface between cell annotation, atlas integration is, clearly massive. And I can see the question asking from Diane Saunders, asking whether the individual bionetworks should arrive at consensus or would be encouraged to arrive at consensus cell annotations that are periodically updated. And that's exactly how I see it, exactly as she's asking. So there's definitely a sort of positive feedback loop, if you like, kind of between, integration across data sets, cell annotation, basically, of that integrated data, and consensus annotation across the biomedical experts within a given network, and then periodic updating. And I think the lung bio network, in collaboration with the analysis working group, has really pioneered that in a fabulous way. I mean, Fabian can maybe describe how that's worked, and Chloe is sort of spearheading a similar thing for PBMCs for blood cells, and obviously also developing and using the cell annotation platform for that. And so that's really how we envision this to move ahead. And when we say integration, it doesn't necessarily have to mean kind of purely automated integration of all data and all cell compartments. Again, there can be a kind of iterative guided, integration of individual cell compartments. Sometimes it makes more sense to divide the data set and separate, fibroblast or stromal compartment from epithelial, from immune, from vascular, et cetera, and then tackle each one sort of compartment by compartment. But that's kind of the sort of iterative way in which we'll arrive at an assembly and a comprehensive annotation of the human cell atlas data. And I'll hand over to Chloe to pick that up and give more color to that, if you're able to, Chloe.
Sure. I mean, I guess the key word that both you and Aviva mentioned is iterative process, right? And so it's a process of going back and forth between integrating additional data sets, revising the annotation, and everything will be timestamped. Because of course, as we add more and more data sets, we're going to get to more granular definition, and more data sets, and I should say more and more modalities. We're going to get to a more granular definition of cell type and states. And in the case of, I can speak for the immune cells, it's not black and white, they're part of transitional states. So the more we add modalities and data points, the better we'll be at defining them. It's also a community-driven effort where we need to engage a different domain experts, which will be distinct across a different organ system and bio network. They need to work together to give feedback on how we're annotating these cells. And this is where the CAP platform comes into play as a tool for all of you to do this. I can speak as an example of how we're proceeding for the immune bio network. We're gearing up to launch cell annotation jamboree this hasn't been launched yet. It's going to be launched in early 2023, and anybody who's interested can join. We're going to be using the integrated object of the first 10 million cells that Aviv showed in the slides in the ROMA that's currently in the QC. And then team will be invited to actually annotate very defined cells in a data object. And then we're going to start comparing notes about how we annotate cells to try to have a conversation around consistent annotation using the same data sets and leveraging CAP to compile our results and conveying this to the general community. So iterative process and time step, and knowing that nothing is final for a while.
Brilliant.
I do also actually want to ask about the other part of Diane's question, because I think we can clarify a point about the process here. Aviva talked about the initial sort of pilot networks that we are looking at, but maybe, Fabian, would you mind maybe talking about the plans for other networks, and what would be your advice to the other networks as we are using these pilots to kind of sort out the process?
As always, and I think how this whole consortium is set up, this is a bit of a dynamic thing, and I think it's actually turning out to be really nice. And this initiative to now do this integration more systematically across bionetworks has been really getting us together and think, and then first take out of the box this process that we've been a bit fine-tuning on the lung atlas, trying out different ones, and as Aviv very correctly said, learning that one size doesn't fit all and we need to adapt. So where things working out very well is with immune system, and starting with kidney and colon, sort of getting there. But for the other ones, we're in contact with all the bionetwork leads, and we said we need to start with the first half a dozen or so, those that we just named, then we continue with the others. But there's already within some of these bionetworks integration efforts happening, and there's then complete game changer type of topics such as developmental atlas, right? This is sort of the whole thing that we're just trying to do organ by organ with an additional time component. So that's complex by itself. And there was just an HCA development atlas meeting in Paris that in particular Mas Hanifa, and of course many of us here have been very active, and just thinking there about merging a few things, reflecting this on top of the adult, or maybe projecting then even organoid atlas, a second whole game changer type of thing on top of that, will need more development. The various groups are thinking about it. There's actually, I think, very ongoing, very lively discussion about it. And what I really liked is that we had a face-to-face in-person kickoff at Vienna last time, and I'm looking forward to do the same at the next face-to-face meeting. But of course, meanwhile, sort of pushing that forward.
And Chloe, do you want to add to that?
Just something very, very quick, right? And so it's hard to integrate all existing data sets in this first draft. So I can speak for the immune cells, right? Some of you may wonder why we went after just the blood for the first effort, and we didn't include, for example, bone marrow, lymphoid organs, immune cells across all tissue. We need to start somewhere with some proof of principle and work out the kinks of how we approach integration and annotation before we start adding layers. And that too will impact how we think about annotation as we start, adding immune cells across different tissues. And so that lesson applies to our other, organ system that have, different ways of sampling tissue and different cohorts and so forth. So.
That's great, thanks. There's a really interesting question about lessons from COVID-19, and HCA has, despite having a focus on healthy reference atlases, has done a lot of work on COVID-19. I was wondering if any of you could draw on those lessons to discuss lessons for other disease studies using HCA data. Maybe Aviv, do you want to start with that?
I'll start with two lessons. I think many here have, it was a very dynamic experience that we all participated in, so I'm sure many will have to comment. First, I'll say that even historically, we can say that the lung atlas integration that we have now actually descends from an earlier effort that was done for COVID-19. So when the pandemic started, really this community rallied amazingly well. People shared data immediately along with necessary metadata of a narrowed kind that we asked for because time was short. And that allowed us to do the first meta-analysis across HCA. It was actually the first, not for COVID-19, it was the first period. and much of the statistics and approaches were invented kind of on the fly, and they were rudimentary at times. But once we got through that, it became very clear, oh, then we can do it for the lung as a whole. It doesn't have to be just for this narrow case. It can be done across the board. And it's true that it was the ambition even from before COVID. We actually had a lung bio network and a seed grant and all of those things, but kind of seeing it in action and happening like on, happening in this accelerated way in 2 months, what normally would have taken 2 years, and people would have gotten together, and they would have had to discuss, and they would have had to do this and that, and they would have had to find the perfect solution rather than the immediate solution. Once we went through the 2 months and we had the answer, then you're like, oh, then I can actually do it. It's not this pie in the sky, it's a thing. And I think that was extremely empowering, both for going through the lung exercise in its full-fledged, and Fabian should speak to that, and from Sarah and my perspective, from saying, oh, then we're ready to go for everything like that. And that takes more time to rally together, have the resources, and so on. So it was an extremely important moment. Before it, a few months earlier, we were in 2019 in a general meeting, and we were talking about the fact that we would need to integrate and query. But after that live demonstration, it became very clear to people what integrate and query could actually mean, and also it's doable, one way or another. The second big lesson was this community has such power because it gets together, because people trust each other, because they understand each other, they have a shared scientific language, they're also friends. So they can do great things together. We always believed it, and we were already doing great things together, but that was a lot greater. And for me personally, it was very inspiring in the work we've done with Chloe and many, many, many other colleagues, clinicians and biologists and computer scientists in the Boston area, to study autopsies from COVID patients. And again, you could say the deck was stacked against us, like everything was against us. It was the early time of the pandemic. We were all locked down. Things were entirely crazy in the hospitals. And, the samples were horrible. Like, there were every reason to say why this would not work. And yet our approach was, can we make it work? And it's not just us. Other groups like these came up all across the world and did their thing. And I think, again, that was very personally inspiring for everyone, from the very early career to much more established scientists, to see how much can be done and how quickly if you just want to do it and you have the tools and the wherewithal. So that's, for me, the COVID lessons.
Sarah, your thoughts?
Yeah, so I was just gonna add, because there's several questions actually in the chats that relate to disease cell atlases. And I guess, just to make the point that HCA in COVID had two roles. The first role was basically in terms of just the pure healthy reference data, providing guidance as to where the virus can dock. So where are the viral entry receptors in the healthy reference body? And that concept of basically a sort of roadmap of the human body for viral entry factors also applies for genetic variants. It applies for drug targets, and it basically is an illustration of the utility of the human cell atlas data as a reference map, or as a molecular guidebook, basically, for all of these features. So it can tell you, where can viruses enter, basically all over the body. It can tell you where's a drug target expressed all over the body. Equally, where's a rare variant? So there's a question on monogenic disease. Where is that expressed? All over the body in a sort of comprehensive way. And it's this comprehensive reference, healthy reference, that has a utility for disease. But then there's also several questions in the chat in the Q&A on the other aspect where HCA is useful, and that's as a sort of for essentially highlighting or computing the delta for disease. And that's if you have, so in the case of COVID-19, it was basically samples from infected patients, and basically the change in the infected samples relative to the healthy reference atlas, is giving us insight into the mechanisms of the pathology and what changes during the disease. And that's obviously you need the healthy reference to understand exactly what's which cells are being infected, how the tissue microenvironment is changing, and so on during the disease. So the human cell atlas really has two serves two purposes with respect to disease data sets in my mind, and I've sort of outlined both of them. And I hope that that kind of serves to answer several of all the different questions related to monogenic disease, infectious disease, tumors, et cetera, that are in the Q&A.
Yes, that's great. Thank you. I'd like to turn to a question about ancestral diversity that's in the chat. Maybe Chloe, maybe direct this to you initially, because I know certainly in the Immune Network, the Asia Immune Diversity Atlas, which is a flagship project of HCA Asia, is funded by CZI, has been a major contributor to the ancestral diversity. Could you maybe talk a little bit about how, particularly with PBMCs, how that has sort of played out and what the path is forward?
Absolutely. Of course, we aim to create a more globally representative resource to understand disease. So we've been partnering very closely, the Immune Bio Network has been partnering very closely with our colleagues of the Genetic Diversity Bio Network, who are generating a lot of data from PBMCs from different regions, and many are part of the recently funded CZI Ancestry network. So we've done a few things. Of course, we're sharing SOPs to make sure that everybody's sharing data in very similar fashion. But also, as we already are thinking about assembling our atlas, we have integrated, as part of this 15 initial data sets, any data sets that were already available that were from different backgrounds, including 2 important contributions from the Asian Immune Diversity Atlas, also known as ADA. So the way we divide and conquer here is that we agreed to come together, the Immune Bio Network and the Genetic Diversity Bio Network, as a team to pick which data sets we'd be integrating, in this case PBMCs, decide on the QC, decide on the annotation process, derive a consensus annotation frameworks together, so both bio network would be working with the same foundation. And then from there, we will empower our colleagues of the Genetic Diversity Bio Network to do their downstream analysis to better understand the contribution of a wide range of metadata to defining cell types and cell states, while the Immune Bio Network is going to be pushing more the definition of immune cells, and together, at the end of the day, we'll arrive with a final product and synergize in the process. And we welcome additional data sets. We have been meeting with the different members to let them know, how we're proceeding. So of course, the first version of the atlas is, including for blood, is not final. We're going to use this product to then add additional novel data sets that include more diverse populations across all regions of the world.
Thank you. No, that's terrific. Any other comments on sort of the general topic of ancestral diversity? We just have a couple more minutes left here in this session.
I think 2 things to say on ancestry. The first is that we're for sometimes the data there are complex considerations with ancestry, both in prospective studies that we will do in the future to collect correctly, and in the data that we already have to understand its ancestral composition, where it might not have been captured, but could be inferred in different ways, as long as they're ethically approved, consistent with the IRB. So that's, I think, one aspect of it. And the second is that in cells, unlike in, when you're just doing genome sequencing, there is always this combination of the genetic contribution and the environmental contribution. Even for the same individual, their environment changes as they move through life and so on. So analytically, I think this opens up a fascinating questions that we're going to have to resolve over the coming years. And in understanding, both the intra-individual variation and the inter-individual variation for cells is something that's going to come very much to the fore. There were many, many questions on the context of disease. And I think without this lens of ancestry and genetic diversity in individuals, it's going to be very difficult to get fully to the bottom in some of the disease questions that go hand in hand with it.
Absolutely. Thank you. This really brings us to the end of our town hall today. I want to certainly thank the panelists for being here. Thank everybody who has signed in. Thank you for your wonderful questions, your interest. Please, if you have any questions about HCA in the future, please reach out to the executive office. You can always write us at info@humancellatlas.org. But otherwise, I would just like to wish everybody a wonderful day. And again, thank you all for coming. Thank you.
Thank you. Thank you,
Thank
John.
you.
Thank you, everybody.
Thanks,
Happy
bye.
holiday season. Bye.